faculty of kinesiology and recreation management at the University of Manitoba, and that's in Winnipeg. She's also a principal investigator at the Children's Hospital Research Institute of Manitoba. So give you a little bit of background for Dr. Saleem. Her, if I'm correct, her BS, MS, and PhD are all at York University in Toronto, Ontario, Canada, and she did her postdoc at McMaster, which is in Hamilton, Ontario. So her expertise is in molecular and cellular physiology, specializing in mitochondrial metabolism and the extracellular vesicle biology, which is she's going to give us more information on today. And she is passionate about the therapeutic potential of pro-metabolic signaling factors mediated by endurance exercise to promote healthy aging and attenuate chronic metabolism-related disease conditions, such as diet-induced obesity. She's been well-funded. She has grants from the Canadian Foundation for Innovation, an SSHRC, New Frontiers in Research Fund, and research from Research Manitoba, University of Manitoba Children's Hospital Institute of Manitoba Fund. She has a New York Academy of Science Interstellar Initiative and a Japan Agency for Medical Research and Development. She has 28 journal articles, 4 book chapters, 2 editorials, and she has been named to CBC, which is the Canadian Broadcasting Corporation, Manitoba's Future 40 class of 2020, to recognize her achievements and outstanding professional and service contributions to the community. So we're very lucky to have her today talking about extracellular vesicles and the regulation of metabolic capacity. So I'll let you go ahead.
Thank you so much for that wonderful introduction, Jill, and thank you for inviting me to give a talk here today. I'm really excited to share some of the, I guess, as I was saying before people joined, some of the fresh new data that my students have collected in the last few years. So without further ado, just to give you guys an idea of what I will be discussing today, I'm going to start off with a little bit of background in my research training and then link it to extracellular vesicles and exercise, talk about their role in metabolism, and also show that they have the capacity to modulate inflammation and other cellular outcomes in different models, and tie it off with some data on how we can use them as therapeutic delivery vehicles. So as Jill was saying, I did my graduate training pretty much at York. I was there for about 12 years, which kind of showed me that once I like a place, I think I stay there. I used that to show my committee at Manitoba that hire me, I'm going to be here for a long time. I'm just kidding. But yeah, so I did my training at York. I worked with Dr. David Hood. He's a skeletal muscle mitochondrial specialist. From there, I went to McMaster, where I worked a little bit more in aging and mitochondrial metabolism and started to get involved in extracellular vesicles. I took a little bit of break in the middle to get my teaching record up. And so I taught at Humber College, and then started as an assistant professor and principal investigator at University of Manitoba and the Children's Hospital. So this was July 2018, and you know, just like got my lab going in 2019, and then a year later, COVID hits, and feels like it's been 10 years since then, but not been 10 years. We're still doing well despite everything that's happened. So where's Manitoba? First of all, this is where we are, this little red province. I haven't had the chance to travel up north because of COVID. Since I got here, first I was too busy, and then I couldn't. But it has beautiful northern lights. So if you ever get a chance to come to Manitoba, I highly recommend that you visit up north. It has the polar bear capital of the world, if you like to look at that. The campus is beautiful. We have, you know, gorgeous prairie skies, and there's a children's hospital, there's a medical school. So one of the best things about this place is how much collaborative everyone is around you, and they're very, very helpful, and we work together on a lot of different things. So before I get into results or data, actually, I just want to start off with a land acknowledgement, because I am speaking to you from Winnipeg, which is along with, you know, the city and the university, they're located on the original lands of the Anishinaabe, the Cree, the Oji-Cree, Dakota, and Dene peoples, and on the homeland of the Métis Nation. And this land acknowledgement is being made to specify that we respect the treaties that were made on these lands, and we acknowledge the harms of mistakes of the past, and we're going to try and do better moving forward in partnership with our Indigenous communities in the spirit of reconciliation and collaboration. So when I first started in Dr. Hood's lab at York University, I looked at the role of P53, which if anyone has ever taken a cancer course or read anything on cancer or does research with cancer, then this protein is not a stranger to you. It's one of the most important tumor suppressor proteins in our body. If you don't have it, you have almost 100% chance of developing some sort of cancer. So P53 is really important, and it's been studied for decades. But when we started looking at it, we were one of the first labs to look at it in the context of physiology, specifically its role in regulating mitochondrial biogenesis and cell death in skeletal muscle. So we identified that when you don't have P53, forget cancer, physiologically, you have reduced mitochondrial capacity, you have these animals don't run as much voluntarily, and this is the wild type mitochondria. And you can see the mitochondria from the knockout animals looks morphologically impaired. And these mitochondria, when we isolated them and looked at their respiration, we found that they were respiring less. So there was a physiological role for P53, which was really, really exciting because, as I said before, this is a cancer suppressor protein. And here we're showing that it's playing an important role in muscle physiology. And even more exciting was the fact that we found that acute exercise could activate P53 and cause it to move to the mitochondria. So this is a blot of nuclear P53. The transcription factor usually hangs around in the nucleus, but we found with exercise, that sedentary acute exercise post-recovery, we found that P53 levels went down in the nucleus, which didn't make sense, but guess where they went up? In the subsarcolemmal and the intermyofibrillary, the two different mitochondrial pools in skeletal muscle fibers. Both of them had an increase in P53 expression with exercise, acute exercise and recovery in a mouse model. So why is P53 coming out of the nucleus and going into the mitochondria? Well, apparently it was binding to the mitochondrial DNA. We were able to pull it down using chip assays and saw an increase in the amount of P53 bound to mitochondrial DNA. And this was surprising because it's a nuclear transcription factor, but we were finding it bound to mitochondrial DNA. So we did a little bit of in silico analysis and we found putative P53 response elements in the D loop region of the mitochondrial DNA, which is the part that controls transcription from the whole genome. So this was really interesting, and it coincidentally matched our data where we looked at the expression of a gene that's regulated by mitochondrial DNA in wild type animals and P53 knockout. And we found that usually this expression of gene COX-1 mRNA increases with exercise and recovery in regular mice, but when you don't have P53, this increase is nearly abolished. So I spent my master, I think I got obsessed with the protein a little bit, but I spent my master's and my PhD studying how P53 regulates mitochondrial biogenesis and function. We looked at post-translational modifications, its ability to be imported into the mitochondria, how it affects electron transport chain function, and also mitophagy and autophagy. And there was a bunch of publications that came out from that. And I was, as I said, I was in the lab with Dr. David Hood. He's an exercise physiologist. So, there's a lot of studies that were being done in the lab on exercise and aging. So I got the chance to be a part of that as well. And I think at that point, probably somewhere along that journey, I became a firm believer of the beauty of exercise. I mean, you guys are in exercise physiology department, so I don't have to convince you, but it seems that we have to convince everyone else that exercise is amazing and it has all of these systemic effects in the body that no pharmacological agent can really recapitulate. At least I don't think I've seen anything. And I remember I was at a CSEP conference as an undergrad, and we invited Dr. Frank Booth to Toronto, and he stood there and said that in studies, we should stop treating the exercise group as the treatment group and start treating them as the control group, because that is the natural way of being, because humans have evolved to be physically active. And I think Dr. Booth is in the audience, so I hope I quoted him correctly, but those words have stayed with me ever since then. We are made to move. Our bodies are designed to move. We're evolutionarily adapted to exercise, and we should exercise because it has all of these effects, from improving fertility to preventing, hypertension, to countering diabetes and obesity. And yet we do the exact opposite. Even as exercise physiologists and experts in this field are, and especially as academics, a huge part of our daily lives is spent sitting at a desk in meetings and writing. And that's not good. So we need to focus and try and take care of this. And this is, everything that I'm saying is not new. People have known about this for a long, long time. This is a picture from one of the catacombs of the McMaster University Library. And I was sitting there and I found these Lancet journals from the 1800s, and I picked one up at random, and I found this paper and said on the systematic effects of exercise. And the quote here, which is, really struck me, was, "It will be universally admitted that properly regulated exercise is absolutely essential for the preservation of a sound mind and a sound body." And if you look at the date of this paper, it's from 1878. So that's old. So we've known about exercise for a long time. We know it's good for us. We know it has all these important health benefits. And yet our society chooses, for sometimes we're not able to do exercise, but sometimes we choose not to do it. And that's unfortunate, because I think if more people were physically active, we'll have a healthier population. And that's, it's proven in so many epidemiological studies and papers. Now, if just in case, if you really still needed convincing that exercise is really good, I'm going to show you one little study that kind of pulled me in further into the field. And this was done by a senior PhD student in my postdoc lab, in Dr. Ternopolsky's lab. And they used a Polgy mouse model, which is a animal that has a mutation in the gene in the exonucleus domain. So this part of the gene allows this protein to correct mutations in mitochondrial DNA. So this animal has a mutation right in that function. So these, this mouse can replicate mitochondria, but it cannot fix mutations that are acquired. And it's a very well-known model of aging, because these animals, as you can see right here, they start to show all the signs that are associated with human aging, graying of hair and loss of body weight. And phenotypically, they look older, but inside, if you look inside the body, they have all the pathological signs of accelerated aging. So wild type animal, sleek, nice looking young guy. Polgy animal looks woebegone. And this is not fighting, this is just ear clippings to genotype them. So if you've never seen this mouse model, I want you to take a good look at these animals, and I want you to keep this in mind when I show you the video on the next slide. So there's 4 animals on this video, and I'm sure that you guys can see the ones that are not moving are probably Polgy. And if that's what you're thinking, you're 100% correct. These are the Polgy mouse that show all the signs of accelerated aging. These ones are also Polgy animals. They're the same age, the same gender. They're actually litter mates. And the only difference between these guys who didn't move at all while the video was playing and these ones who looked just like regular healthy wild type animals was that these poultry animals exercised for 5 months. So just by exercising, you can see that this transgenic mouse model looks so much more healthier. So, but that begs the question, when you're exercising, your muscles do the work. Well, why is it that the rest of your organ systems and body is adapting? And that links me to the next part of my presentation, which is about the concept of myokines. And really important work in this field has been done by Dr. Bente Petersen of the Copenhagen Muscle Research Center, who established really that muscle is an endocrine organ that can release these myokines. One of the first ones was IL-6, and since then there has been so many more that have been identified. And these myokines can talk to organs, liver, fats, adipocytes, even more that are not shown on this schematic here. And it's not just a one-way conversation. These organs are also releasing proteins and factors that can come and act on the muscle. And also they're talking to each other. So there's a lot of conversation happening. Now, when proteins or transcripts are released into the circulation, they can potentially have a very short half-life because they're not designed to be in the blood. And that is what got us to extracellular vesicles, because this is one of the ways in which many of these proteins can be released from these different organ systems. And this is an evolutionary conserved mechanism of cellular crosstalk. So as long as you are a living organism, it doesn't matter if you're a human being or a potato, you release extracellular vesicles. This is what your system is using to talk to each other. And there are many, many different types of EVs. Traditionally, they were divided into really small exosomes that are 30 to 150 nanometers. This is the size of the SARS-CoV-2 virus that shut down the entire planet for the last year and a half. And these exosomes are formed by an endosomal pathway, or you can have them bud out of the cell, like the microvesicle, which is a little bit larger, 100 to 1,000 nanometers. You also have apoptotic bodies, which are even 5 times as big as microvesicles. And you will see that there's a lot of different nomenclature that is used in the field to refer to them. So the facts about extracellular vesicles, or EVs, as I'm going to call them for the rest of the presentation, is that they are released from every living cell and that they carry biological cargo. And this cargo can include, microRNAs, mRNAs, non-coding RNAs, proteins, lipids, metabolites. Whichever cell is producing the EVs and whatever is the condition of the cell affects the cargo. Now, they found, I think, the first microRNAs or mRNAs inside EVs in the early 2000s, and that's when it was discovered that these EVs could transmit messages between the cells. And since then, research in this field has exploded. And we hypothesized in 2016 that bringing it back to muscle and exercise, that it is possible that a contracting muscle packages myokines within these EVs or exosomes, releases it into the circulation, so it can travel distally and systemically to every organ system. And we were one of the first groups to hypothesize this. And at that time, we knew there was a bunch of known myokines that were found within extracellular vesicles. And since then, there has been a growing interest in the role of EVs with exercise. And I think up to about 400 different protein markers have been shown to be released, packaged within EVs from muscle. And a large majority of them are myokines, which is what I decided to base my research program on. So what I study in my lab is how EVs can transmit on a very basic molecular level messages and participate in cellular and organ crosstalk and regulate my favorite organelle in the world, mitochondria, and regulate its function in different conditions of health and disease. I also have connected this to some clinical studies where we're looking at children with obesity, as well as birth cohort studies and studying the role of EVs in these patient populations. And the really cool stuff, which is the one in the middle that we're just starting to get going in the lab, which is the translational aspect of using whether transgenic mouse models or ex vivo models to study how we can manipulate this capability of EVs to transmit messages and affect cellular function. So the first thing what I'm going to show you is whether EVs can work as the exercise pill within nature. So this in this study here, this is a pilot project that we did, and we took mice that were sedentary and we isolated their serum and we injected it into sedentary mice, or we took mice that were exercised 5 times a week for 8 weeks, standard, treadmill exercise for animals, took their serum out and injected them also into sedentary animals. The first thing I want you to focus on is this graph on the top right, which is time to exhaustion. How long can an animal run on a treadmill? And this is a well-known fact that if you exercise train animals, they will last for a longer time because they have higher endurance capacity. Now, the really cool thing we saw was that when you give sedentary mice serum, or specifically in this case, exosomes from these endurance animals, their time to exhaustion increases, almost matching it to animals that were exercised for about 2 months. And this increase in whole body physiological capacity was matched by changes on a molecular level, where again, we saw that the sedentary animals right here that were given these exosomes from these endurance mice had higher mitochondrial DNA copy number than just regular sedentary mice. And it actually matched the amount of mitochondrial DNA that is found naturally in exercised animals. So mitochondrial DNA copy number being a proxy measurement for mitochondrial content. And there seemed to be an additive effect. So if you took mice that were trained and gave them extra exosomes from other animals that were also exercising, they seem to have an additive effect and they increase their mitochondrial DNA copy number a little bit more. So that was really exciting. And over the past 4 or 5 years, there's been growing interest in EVs and exercise. There's a bunch of papers, and this is not even a comprehensive table. It was a recently published review article that detailed the different studies that have been done to show the effect of acute exercise on circulating EVs. And I put it up here because I wanted to make 2 points. One, that there's a lot of research on it, but the second is that there's a lot of variability in results. If you look here, this paper showed a 2.5 or 2.7-fold increase in the number of EVs after acute exercise. This paper showed no change, and this paper showed no change, and this one showed 2.5-fold increase in particle count. People are using different models: treadmill exercise, submax exercise, in men, in untrained males, in healthy or obese men and women. So there's a lot of heterogeneity in the type of exercise models that are being used. There's differences in the isolation methodologies that are being used, which affects what people are calling them. So some are calling EVs exosome-like vehicles or particles. Some call them EVs. So there's a lot of, if you pick up an EV paper and if you find terminology that doesn't match another paper, don't be surprised. This is a very common problem in the field. Despite that, despite the small discrepancies from papers, overall, it seems the majority of papers are showing that with exercise, EVs seem to be involved in that response, especially in the acute exercise part. But we still don't know how critical they are. Just because they're being released doesn't mean they're important. So this is one of the first studies we did in my lab when I started, we collaborated with Dr. Jonathan McGavick and Dr. Martin Seneschal, and they had published this study linking the amount of irisin, which is a myokine that is released from muscle, a priori to exercise training, and they tried to evaluate whether it could predict who would respond to exercise. So the model they used was young adults around 15.5 years of age. They did an acute exercise intervention. They took blood at this point and they measured irisin levels. Then these same subjects were put on a 6-week training program, and then they measured, changes in VO2 and insulin sensitivity. And then they tried to see if the a priori release of irisin could predict who adapted to exercise 6 weeks later. And what they found was that basically the more irisin you released before training started, the higher chance you had, or the higher propensity to improve your insulin sensitivity as measured by the Matsuda index. So in this study, our collaborators found that irisin release before training could predict who responded to training. So we thought, why don't we look and see if EVs can do something similar? So this is work that was done by my postdoctoral fellow, Dr. Tana Piardona, as well as a high school student who joined the lab in grade 11, came back in grade 12, and came back for the 3rd year now in first year university. So start them young and keep them interested. But she did a phenomenal job on this project with the postdoctoral fellow, and that's Alexandria. So we isolated EVs from the same cohort of subjects, young adults with obesity, before exercise training. We used size exclusion chromatography, which is one of the best ways to isolate EVs from plasma. Not the only way, there's other ways as I'll allude to afterwards. And we characterize them. So when you work with EVs, the first thing you have to do is look at their size, their properties, their biophysical properties, and the marker proteins. And I'll show you a couple of data points from this study. So the subjects that responded to exercise in pink are called RE, or responders, released EVs that were around 150 to 300 nanometers of size, whereas the non-responders primarily had a enrichment of the smaller EV population in their plasma. And another interesting thing we found was that during exercise, so this is acute exercise, 0, 15, 30, and 45 minutes. This is 120 after recovery. We looked at the amount of EVs that were released in the serum of these subjects, the non-responders in black and the responders in pink. And we found that those who adapted to training were producing EVs that had more protein in it during exercise. We don't know what proteins were packaged within the EVs. We don't know what the function of those proteins, but clearly there was a significant difference between the two groups. And I guess most relevantly, the size of EVs, we found it to be correlated, not completely significant because of this very small sample size, but it seemed to predict who would respond to exercise training, just like our collaborators found in the case of irisin and insulin sensitivity. So we clearly have to do more work here to identify, what's inside these EVs and where are these EVs originating from? And, if we increase this was an N of 4 or 5 per group. So if we increase the N size, does this become more significant? Now, that was acute exercise. What about chronic exercise? Now, the number of chronic exercise studies that have been done with EVs, you can count them on one hand, and shown on the slide is a summary of them. There's about 5 so far. And all of them have been done in mice or rat models. And again, I put this table up here to show you the heterogeneity in the different models that have been used so far, which kind of confounds the results sometimes. So people have used, swimming or treadmill exercise. Majority of the groups looked at plasma EVs, and they found, again, an increase in EV count mostly, consistently, although some papers reported there was no change. Now, when you look at plasma EV population, it is very hard to find skeletal muscle-derived EVs in that isolations, because the predominant fraction, according to the papers that have looked at it so far, report that EVs in the plasma originate from endothelial cells or reticulocytes or white blood cells. Now, this has to be taken with the caveat that people are trying to determine the origin of EVs from skeletal muscle by using a marker for skeletal muscle, which is alpha sarcoglycan. The problem is that not all EVs from muscle would have that marker. So the reports that suggest that the plasma population of EVs that comes from skeletal muscle is very small has to be taken in that light, because we don't actually have the best purified, most reproducible method of determining the origin of the EVs. So this necessitates using an in vitro model, which is what I've done, or what we have done in our lab, where we used C2C12 mouse myoblasts, and we are now replicating this with human cells as well, and using in vitro models of acute exercise and chronic exercise, isolating the EVs and looking at organ crosstalk between other relevant metabolic tissues. And we started off with skeletal muscle to see if muscle-derived EVs can, have a autocrine function and affect other muscles. But there's a study ongoing in the lab that's also looking at cancer cells. And I'll come back to that in a couple of slides. So the first thing we did was look at acute exercise. So these are mouse myoblasts. We differentiated them into myotubes, and we found that, with differentiation, there is a change in the EV population, and this actually matches a number of papers that have looked at that already. And this work was done by my master's student, Ben Videk. We looked at the expression of exosomes and microvesicle proteins, so ALX, TSG101, CD63, CD81. These are all markers of exosomes. We found them to be higher in myoblasts and lower in myotube, which kind of didn't make sense, because overall we found myotube EVs to be smaller. But this is the data that we had, and we found microvesicle markers, which are associated with bigger EVs, to be more highly expressed in myotubes. So clearly an effect of differentiation there. So then we took those myotubes and we acutely stimulated them by using an electronic pacer, and we looked at size, really no difference. Zeta potential, which is the stability of EVs. So the more stable the EVs are, that means the less they're likely to aggregate. And if they're not aggregating, they're more functional. So zeta potential values are usually negatives, and we saw, both control and stim were comparable. No difference in the protein yield between the two. And here's another look at the different size of the EVs, and control is in light blue and dark blue is the acutely stimulated myotubes. And again, pretty much the exact same. So we decided that we're going to go and then look at chronic exercise as a model. So again, using first mouse skeletal muscle cells differentiated to form myotubes, being paced for 3 hours a day for 4 days, which is a very well-established in vitro model that mimics chronic exercise. We isolated EVs, and this work is being done in the lab by my PhD student patient, who's being held by my tech, Samira. And now, instead of using plasma, the size exclusion chromatography method of EV isolation that we use for plasma, we decided to use DUC, which is differential ultracentrifugation. It's one of the gold standard methods of EV isolation, and it works really well with cell culture as opposed to using the SEC method with the plasma. This is what EVs look like if you were to isolate them and do an EM analysis, and this is the expected size, and you can see the nice cup-shaped appearance, which is classic of small exosomes. So we knew we were getting purified and intact and not aggregated isolations from our model. And with chronic exercise, we saw that there was a lot of things going on here. So first, average EV size was lower in the chronically stimulated myotubes, which is in dark blue. Zeta potential with the stability, while both were pretty negative, the CS, the chronically stimulated myotubes, had a even lower zeta potential, so the EVs were even more stable. The protein, again, was indifferent between the two. And the size distribution, which is the different EV sizes here on the x-axis, black is control and blue is chronic stim. You can see that with stimulation, with chronic stimulation, there's a propensity to have higher 150 nanometer size particles, which is the size of exosomes, whereas the control groups had more of the larger particles. So does this have any significance? So we started to do co-culture experiments, and the model we use are, again, our in vitro model. We take cells and we treat them with 3 different conditions. Conditioned media from either control or chronically stimulated myotubes, just the skeletal muscle derived EVs, or the EV depleted media to see if there's maybe something else outside of EVs that could have an important effect on our outcome variables, which is, first of all, mitochondrial biogenesis and metabolic capacity, and also looking at cell death and viability. And we started off with looking at muscle-to-muscle crosstalk, but before we do anything, we had to make sure that EVs were actually being taken up by the cells. So we did some confocal microscopy imaging. EVs here are labeled with a green dye. This is red in F-actin, and this is a zoom or a magnified image, and you can see this beautiful, proof of EVs being taken up by all the cells inside. And this was important for us to know, because if EVs are stuck on the outside of the cell, then we can't really expect a function, or, well, we shouldn't really expect changes in cellular function, but the uptake mechanisms or how these EVs got taken up into the cells, we don't know that yet. And I put this picture here to show the complexity of the different ways in which EVs can be taken up. They can be endocytosed, or they can bind to a receptor and then be endocytosed, or they can be taken up by clathrin or calveolin mediated pathways. They can be phagocytosed or micropinocytosed, or they can just fuse directly to the membrane. And the only way in which they can be outside and still affect cell function is if there's a receptor that they can bind to and cause or trigger a cascade of events that can change cellular function. We don't know which one of these mechanisms was activated in our model, but at least we know that EVs got inside. But we hope to establish how they got inside in future. So what happened once they got inside? Well, first, if you focus here to the left, what we did was we stimulated the myotubes and we isolated EVs after each bout to mimic basically the repeated bouts of a chronic exercise model. And each time cells were treated with conditioned media, which is CM, EVs, or the EV depleted media. And the first thing I want you to look at is COX activity, which is a marker of mitochondrial content. And we didn't really see any significance in the conditioned media or the EV depleted condition, but the EV treatment condition had a significant increase in cytochrome C oxidase activity, which is, you can see it in isolation over here. And this matched another method that we used to look at mitochondrial content, which is mitotracker staining of the cells that were treated with the EV. So they both increased in the EV treatment group. In addition, we found that there was an increase in cell count just in the EV treatment group, not in the conditioned media or the EV depleted, and that all of these things might be happening at in a very cell-specific manner, because when we did the same thing with lung cancer cells, we saw a completely opposite effect. So if you focus here where my pointer is, this is EV in blue, decreasing the viability of cancer cells, whereas they had the complete opposite effect in physiologically relevant skeletal muscle cells. So we're still looking to this. This is we've done the preliminary work with the muscle cells, but we're starting to look into cancer cells with a little bit more in-depth fashion. And this, I'm really, really excited about this because when I was doing my PhD and I was writing my comprehensive exams, I found this paper from 1962 by Hoffman et al., who took rat muscle fibers, stimulated them in a isolated bath, took that bath, lyophilized it, and injected it into tumors, and found that every single tumor that was injected with that bath coming from that exercised muscle fiber completely disappeared or was reduced. And this is from 1962. They called it the F factor. And since then, I haven't seen anyone follow up on what was that F factor that was released from these muscle fibers that completely, ameliorated the tumor size. So I really hope it's the EVs. That's my hypothesis, but I'm excited to see where our data takes us. So I hope I've shown you by now that chronic exercise-induced skeletal muscle EVs can affect metabolism, but also other variables in a very cell-specific manner. And this is not just related to exercise-induced release of EVs. We find this ability of EVs to modulate cell function in other biofluids as well. So another project that's going on in the lab is looking at EVs in human breast milk and seeing it linked with asthma. Now, remember I said EVs are produced by all cells and all living organisms produce them. They're in every biofluid, so they're also in breast milk. And we know breast milk is really good for early child development. Mothers who breastfeed their children, those children have a lower incidence of asthma. So this is a project that spawned off with that collaboration with Dr. Azad and the child cohort study, where we tried to look at EVs from these mothers who had asthma versus who didn't. And these EVs were isolated from breast milk, and then we tried to link it to the asthma incidence in the offspring. And I won't go over what asthma is, but we isolated the EVs from breast milk using, again, size exclusion chromatography. And these females were pretty well matched in terms of age, BMI, and their ethnicity. And the only difference between the control and the treated group was the presence of asthma in the mothers. So again, this is how we isolated the EVs. And this work was done again by my postdoc and the high school student who was helping her during this part. And I'm just going to zip through this work here. The first thing we did was we characterized these EVs, and we found them to be, we took the fractions that were enriched in small EVs or exosomes, and we found that the size of EVs was smaller in the breast milk from mothers with asthma, and they had 7-fold higher particle count in the breast milk. We did a proteomic screen on it, and this is literally me making the pathway analysis last night. We found that 40 proteins were downregulated, 44 upregulated in the mothers with asthma. And these proteins are linked with different pathways. And what was really exciting is that they're linked with the inflammatory or the immune system pathway, which bears direct relevance to co-culture studies that we did in the lab, where we took human airway smooth muscle cells and we added EVs from mothers with and without asthma to it. And we looked at the interleukin-6 secretion, so the pro-inflammatory cytokine secretion. And we treated these cells with, for time course experiments and with and without the presence of interleukin-1 beta, which stimulates an inflammatory response. And these cells, we obtained these cells from our collaborator, Dr. Haleko, who has the world's largest repository of human airway smooth muscle cells. So again, first thing we wanted to do was that EVs get taken up by these smooth muscle cells. So we labeled them, put them on the cells, and we saw that all the both the EVs from the control mothers and the mothers with asthma were being taken up. But the most important part is that the mothers who had asthma, their EVs were actually anti-inflammatory in the sense that they were decreasing the amount of IL-6 released in control conditions. So EVs are not just regulating metabolism, they can regulate different pathways within the cell. And this bears this bears physiological importance because we don't know if this is a protective effect or if this is a causative effect in the development of the child who is drinking this breast milk, and whether or not they will develop asthma or whether they will be protected from asthma. We don't know that yet, but this is really exciting stuff. So, so far, what I've shown you is that chronic exercise-induced skeletal muscle EVs can affect metabolism, and that EVs can also affect other cellular functions like inflammation. And can we manipulate this ability of EVs to change cell function? Yes, we can. So we can take these EVs and we can package really anything we want in it. So this is an example of EVs being packaged with Duchenne muscular dystrophy, a dystrophin mRNA, which is deleted in Duchenne muscular dystrophy. And what we did was we took wild type animals and we looked at dystrophin expression, and that's the red dye here. And dystrophin is the protein that kind of holds up the muscle fiber. So if you have, imagine a poster stuck on a wall and you have those pins that are holding up the poster. You take away the pins, the poster falls down. So dystrophin kind of works like that push pin on a poster. If you don't have dystrophin, the muscle collapses. If you have dystrophin, it maintains its structure and its morphological shape. So dystrophin is really important. MDX mice are mice that don't have dystrophin. You can see that there's no red protein expression. And when we package EVs with the dystrophin mRNA and inject it into animals, we can rescue the expression beautifully. And this rescue of the expression is linked with a rescue in function. So these are the animals injected with the dystrophin mRNA packaged in exosomes, and this is muscle grip strength, improved over the knockouts, but not back up to wild type levels. So EVs reflect the cellular milieu of the parent cells, whether they're coming from sedentary cells or exercise cells. It changes their cargo. We don't know how they're released, what causes them to be released. Is it calcium signaling pathways? We don't know how they're specifically taken up. We know that EVs have the capacity to modulate cell function. So they can affect metabolism, they can affect the inflammatory profile. And they clearly seem to do it on a very cell type specific manner. So lung cancer cell count went down, but muscle cell count went up. How did the EVs know which cell was which? It was the same EVs being put on both cell types. We don't know that yet. And clearly we can use them to deliver biomolecular cargo. So can we package EVs with the one protein that increases, metabolic capacity and give it to people who can't exercise? Can these injected EVs, if we give them orally, can they cross the gut intestinal barrier? Can the breast milk EVs from the mother with asthma or without asthma cross the intestinal barrier? Those are all questions that we don't know the answers to, but I hope that my lab can hopefully generate data and some results to eventually answer them. So I'm going to stop there and thank you all for listening and for attending this talk. And I know I kind of added a little bit extra stuff at the end, but it was really cool data and I wanted to share it with you all. But none of it would be possible without excellent trainees in the lab, and the underlying names are the ones whose data I showed today, and all of our collaborators and funding agencies. And I'd be very happy to take any questions at this point.
Well, thank you so much. We're going to clap for you in the end, but I'm clapping for you now because it's such a fantastic presentation.
Thank you.
And it was also interesting, and most of it, but because I do clinical work and translation work, so I appreciated that, the studies that you presented at the end. And Very
I'm
interesting.
glad. I feel like I zipped through them because I wanted to leave 10 minutes for question answers.
No, that was perfect. Maybe we need to have you come in for another talk and just on that. Okay, so we have a lot of questions in the chat window.
Okay.
Would you like me to read them, or are you able to
Sure.
find them? Okay.
I cannot see my chat box right now.
Okay.
Oh.
All right. So a question from James. One of my main research projects right now is looking at how EVs carrying microRNAs from the liver are regulated during chronic exercise. How would you compare liver EVs output to skeletal muscle?
Okay, so that's a great question. And again, I kind of alluded to it in the presentation. When you're looking at systemic EVs or EVs from plasma or blood, we're dependent on using a marker protein to identify which proteins originated from a specific organ. And depending on how good that marker is, you can identify all of the EVs that came from it, or you can identify 50% or 40%, and we don't know that yet, at least not for every organ system. So that's one thing, how specific your marker is for your organ. And how do you know that every EV that came from that organ of interest is packaged with that same marker? That's also another thing that we're not sure about. So until we, what I would advise is to start off with what we're doing in the lab, which is using in vitro models first to establish. And then there's some really beautiful transgenic animal models that you can use where you can have a liver specific Cree and use that mouse model and isolate EVs that have Cree. Hopefully it will be better than using just a regular protein marker for liver, for example, and then use that marker and use it in another model to see liver EV uptake in other organ tissues. That would be my advice. But I'm sure there are better methods, and someone else can probably talk about it.
Thank you. Next, Dr. Parks had a question.
Go ahead and read it.
Okay, can you expand a little on the markers that are used to verify EV isolation purity?
Yes, absolutely. So the minimal information of EV studies is a 2018 paper that was published by the International Society for Extracellular Vesicles, and they mandated what we have to do every time we do EV research. So size, zeta, and then looking at different marker proteins. And that's why, every study has that characterization panel. So you have to look at markers that are related to small EVs or exosomes. We have to look at markers that are related to microvesicles. We also have to look at markers that are lipoprotein specific to see the purity of our isolations and if there's contamination by non-EV co-isolated proteins. Then there's, depending on what you want to do, there's a huge table that they try and say the more you do it, the better it is. Obviously, it takes a lot of time to run all those Westerns and the proteins and antibodies, but microproteins, size characteristics, data potential, and then electron microscopy imaging. Those are like the 4 basic minimum criteria that you have to meet. And the importance of doing that is because of what I was showing with the heterogeneity and how people are using different terminologies and different methodologies to isolate it, and they're calling them different names. And there's a lot of it confounds the main point of EV research, and people start losing forest for the trees. So they're trying to standardize some of those. So for purity, we're definitely using the lipoprotein and looking at EMs.
That's what I thought. Yeah. Thank you for that. And it's very cool to watch somebody in a relatively early field, because it's a really challenging time to be an early career person in an early field. So that's really neat. I'm going to ask my other question really quickly. Those pictures are just wonderful because they just speak 1,000 words to me. It looks like there's a hazy ring around. Is there a glycocalyx or is that just something from the photo around the edge, around the outer edge?
This is the electron microscopy image?
Yeah.
Okay, let me just quickly put it up on the screen. So I think, where is it?
It's down. And
there's one here. So this little halo,
Yeah, it looks like there's some gooiness, yeah, around the outside.
Yeah. So this is an artifact of electron microscopy.
Okay.
So, but it is a classical way of telling the EVs because they have this cup-shaped appearance. But this cup-shaped appearance disappears when you look at cryo-EM microscopy.
Oh, yeah.
So if you look at cryo-EMs, you see them more in their native form, and they're, heterogeneous in terms of size, but they're all round, and you don't have that cup-shaped appearance.
Yeah, thank you so much.
You're welcome. Thank you. Thank you for your questions.
Okay, Dr. Padilla asked, if you experimentally prevent the release of EVs, do you preclude exercise training induced beneficial metabolic adaptations?
Theoretically, yes, absolutely. And again, great question. It depends on, I guess, the method by which you stop the EV release and how specific that antagonist is. So there is a couple of EV inhibitors. One is called Dinosaur, which is my favorite because it's called Dinosaur. So I'm going to use it, but they're affecting other cellular pathways that will be affecting endosomal sorting and escort proteins. And those are involved in so many other different processes as well. So my fear with that is that you can use them, but then how do you know A, it's 100% effective, and B, what about the off-target effects? So until we know, of a good specific inhibitor that only suppresses EV release and doesn't affect anything else, I think we have to keep that in mind. But those are absolutely very viable approaches to stop EV release and then exercise and see if we get the same adaptations, for example.
Thank you, and very excellent presentation. I really enjoyed it.
Thank you very much for your question. Thank you.
Okay, it's a question from Dr. Vera Potter. She mentions that the adipocytes kept popping up on your slides, but she would like some more information about the role that they play. And any thoughts on exosomes from other tissues, example, muscle, ending up in and affecting adipose tissue? Or what about those exosomes being released from adipose tissue?
So great question. And I do want to work on adipocytes. That's why they're there. We started working on 3T3 cells, and then COVID kind of changed everything. And then I lost my postdoc finished her work, so we couldn't get back to it. But let me see if I have that data here. We did some pilot work with mice that had Cree package in their EVs. And we took those EVs and we injected it into another transgenic animal. These are Rosa mice. So Rosa mice have, let me just share this. So Rosa mice are reporter mice, and usually they express tomato red, but that's a flux gene. And if you add Cree-containing EVs, that red gets deleted, and then you get a switch from red to green fluorescence. So this is one of those transgenic mouse models I was saying. And what we did here was we took EVs that were packaged with Cree, and we injected it into Rosa mice, and we could see the red to green fluorescence change in eye, in the colon. And when we looked at mRNA expression of EGFP, we found them in liver, in epididymal white adipose tissue, inguinal white adipose tissue, colon, eye, brain, even brain, because EVs can cross the blood-brain barrier, as well as in the testes. So definitely there is ability of these different organ systems to take these EVs up, and adipocytes is probably the next thing you want to go to once muscle and cancer is done. We took some EVs, and I don't have those slides to show, but we took some EVs from our chronically stimulated group, and we put them on 3T3 cells, and they had smaller lipid droplets. But that was an N of 3. It was just like let's just try it out and see what happens. But we have to really, really look at that for sure.
Super cool. Thank you so much for that. And great talk. I really enjoyed it, and great data also.
Thank you. Thank you so much. I really appreciate that.
Yeah, I have one last question. Would you expect resistance training to have a greater impact on skeletal muscle derived EVs compared to aerobic training?
That's a great question. So I'm an endurance exercise person. So I want the endurance exercise to have the better effect, but There's some, I think I saw a paper that looked at resistance exercise and they were looking at EVs and I can't remember what they found. No one has done these comparison studies yet, but it definitely would make sense if the muscle is activated for it to be involved in releasing myokines or EVs rather. But yeah, I'm not sure. My heart says endurance exercise, but that's just a guess and a wish.
I guess that's a good idea for another study.
Do you think the release is intensity driven?
Yeah, intensity driven, time of day driven, the model of exercise that you're going to use, where you're going to isolate it from, also methodology driven, which methodology to take to isolate it. So yeah, lots of different variables to play with.
Great talk.
Thank you very much.
Well, thank you so much. And if everybody could just unmute their mics and give Dr. Saleem a round of applause.
Oh, thank you guys.
Thank you.
Hope you can hear us.
We had other questions in the chat, but I can just email those to you.
Sure, absolutely. And if you have any questions, please reach out, and I'd love to grow and collaborate and look at different models and different systems, because we need to do this work. And I think EVs are really, really cool, and we need all the exercise physiologists to join. So thank you again for inviting me. I had a blast, and thank you for the excellent questions.
Thank you
so much. Have a good day.
Thank you.
Thank you.
Bye-bye.
Bye.
Thank you.